Hello, I'm Patrick Shaw-Stewart, and I was asked by the South African COVID Environment Reference Group to make a presentation about the seasonality of respiratory viruses and how that might affect COVID-19. And I thought I would make a video of the presentation. I'm going to try to convince you in this talk that we should focus on temperature if we want to understand both the seasonality and the pathogenicity of respiratory viruses. And this analysis can be applied at many different levels, from the level of the individual patient to the biochemistry wet lab. So this is a graph showing what I mean by seasonality. You can see here that every year a whole lot of different respiratory viruses become active during the colder 6 months. At the bottom I've printed temperature. Every time the temperature dips, you can see that there's a peak in the number of respiratory illnesses, and this applies to really almost all respiratory viruses. And you can see that you can't really generalize about what time of year particular viruses come. They just tend to come at some point during the colder season. So, for example, RSV sometimes comes early, as you can see here, but sometimes it comes later in the season. Now, there are many different viruses that cause respiratory illness. There are at least 200 different strains, and there are many different unrelated viruses including in those. So, for example, there are double-stranded DNA viruses, there are RNA viruses, positive sense, negative sense. Some of them have lipid envelopes, others don't. So they're really as different as they could be, but they all have in common this winter seasonality. They all cause more illness in the colder 6 months of the year. Now, other viruses such as polio and rabies have summer seasonality, but they're not respiratory viruses. And in fact, the only exception that I know of is one particular respiratory virus, which isn't very common, called parainfluenza type 3, and it's more common in the warmer 6 months of the year. Now, there's really no good explanation for this seasonality, and I would maintain that if we can't explain seasonality, we don't really understand the illness. And if we could explain it, it would probably help to control epidemics and also develop vaccines and drugs for respiratory illnesses. So, what are the conventional explanations? Well, the explanations that you'll see in the scientific literature are based around 3 basic ideas, and I've called them M1, M2 and M3. M1 is that we crowd together more in the winter. M2 says that the virus survives outside the body for longer in the winter. And M3 says that in some way our immune defences are weaker in the respiratory tract in particular in the winter. But all of these explanations have a problem, and that is that if you look in the tropics, all the viruses, which are virtually the same strains that we get in temperate areas, are present year-round in the tropics, and they're there at intermediate levels. Whereas in temperate areas, most of the viruses seem to virtually disappear in the summer, whereas they're very prevalent in the winter. So, for example, if you look at M2, it's often said that influenza is more stable in cold, dry conditions, but rhinovirus prefers cold, damp conditions. And the difficulty is that both of them thrive in the tropics year-round, and very interestingly, both of them arrive in the rainy season in places in the tropics that have a rainy season. So this is the exact opposite of what this explanation predicts for influenza. It shouldn't survive in hot places, and it certainly shouldn't survive in hot, wet places. So I'm going to show you some data here which shows the prevalence of influenza, and the authors have plotted the number of people who get sick from influenza in 4 places around the world. And you can see the seasonality in North America, in Bismarck, North Dakota, where the virus seems to disappear in the summer, and then it's there again and becomes apparent in much greater numbers in the winter. In Singapore, it's there at intermediate levels all the year round, and you can see that it tends to come during the rainy season. Wind and rain both cause people to become chilled, and they both seem to increase the number of cases of colds and flu that people get. So these 2 peaks in Singapore correspond to the southeastern monsoon and the northwestern monsoon. And in Fortaleza down here, I've plotted rainfall, and it's very clear that there's a very strong correlation between the rainy season and influenza and other respiratory illnesses. So, what's my explanation? Well, I've proposed M4, and this says that respiratory viruses are temperature sensitive, and they use temperature to keep out of the lung, heart, brain, etc., because if they infect those organs, they're going to immobilize their host. Now, there's also quite a well-known hypothesis in evolutionary biology called the transmission virulence trade-off hypothesis, and I'm going to illustrate this by taking two extreme cases. So on the left, I'm looking at chickens in a barn when bird flu gets into the barn. On the right, I'm looking at the wandering albatross, which is a bird that spends months at sea on its own in the Southern Ocean. So if you think about first the chickens in the barn, if the bird flu gets into the barn, no bird can escape exposure because the birds are all crowded together. And what tends to happen is that the virus can be present in the barn for a long period, but if it begins to mutate and becomes a bit more active, then it can suddenly, very rapidly, become very, very virulent, and it may well kill every single bird in the barn. And what happens is that the fastest growing strains are selected because they arrive quicker, so they can outcompete the slow growing strains. And it doesn't matter to the virus if the birds are immobilized or if shedding is only short-lived, because there are so many opportunities for transmission. So if the host dies, but the pathogen has already been spread to another 5 or 10 birds, then that strain is still going to be selected. And in these conditions, greater viral activity and greater virulence is favoured. Now, if you think about the wandering albatross, the birds are going to be alone for weeks on end, so shedding now needs to be long-lasting because the bird needs to be shedding virus when it meets another member of its own species. If there was a virus that was as aggressive as the bird flu in the chicken barn, then the albatross would fall into the ocean and die, and that would be the end of the virus and the bird. So in this kind of situation, it's very important to the virus that the host isn't immobilized, and generally speaking, milder strains tend to be favoured in those conditions. Now, the virus wants to have its cake and eat it. It wants to shed as many virions as possible in order to transmit itself for as long as possible. And one way that a respiratory virus can do this, in mammals and in fact in birds, is to develop temperature sensitivity, because the nose and throat are some of the coldest parts of the body, and if the virus just refrains from replicating and developing in the warmer parts of the body, then it won't immobilize its host, but it can cause an aggressive infection of the nose and throat, which allows it to be transmitted to other hosts. And if you think about what's happening in COVID, the lockdown makes us a little bit more like the albatross because we're less likely to meet other potential hosts if we're carrying the disease. And also, if herd immunity increases, that also makes us more like the albatross because, once again, an individual who's infectious is then less likely to meet a susceptible individual. Now, I'd like to emphasize that this hypothesis is not normally about how long the host survives. Selection is not normally driven by whether the host dies before it can transmit. It's about how long the host is infectious for. And generally speaking, viruses that cause their host to be infectious for a long period tend to make us less sick because the virus has to be less aggressive, it has to replicate slower, and it has to moderate its activity. Viruses that develop very rapidly tend to make us more sick, which is intuitive. So I've called this hypothesis that most respiratory viruses use temperature to confine themselves to the nose and throat the TDVT hypothesis, and that stands for the temperature-dependent viral tropism hypothesis. As I said, we need to think about the fact that the respiratory tract is one of the coldest parts of the body, and in fact, if we're chilled, for example, if someone puts a wet towel on our back, the temperature in the nose and throat drops further very rapidly. This was actually shown by an American doctor whose name was Mudd, spelt M-U-D-D, in 1919. And there's actually a gradient of temperature from the nose down into the lungs. So we have the idea that it helps the virus if it can confine itself to the upper parts of the respiratory tract. And it seems that almost all of them use temperature to do this. And the reason I say that is that they all have this slightly mysterious seasonality. And I believe that the seasonality of respiratory illness is a side effect of this mechanism, which is really designed just to allow the virus to transmit itself without making the host too sick. Now, you'll see that another cold part of the body is the toes, and I'm going to come to that later on. So, how does this work out in practice? Well, I'm going to show you a graph here that was published in 1958 by Edgar Hope Simpson, who was an English doctor who was very interested in colds and flu. And he's plotted temperature together with the morbidity of colds, that's to say, the number of people who had colds in his medical practice in Cirencester in the UK in 1954 and 1955. And he's inverted temperature. In other words, he's plotted high temperature at the bottom of the graph and low temperature at the top of the graph. And I'm going to start with the situation in the summer. So the virus has to have relatively less temperature sensitivity because the air is warm, so the nose and throat are relatively warm. And if it was very temperature sensitive, it would be completely inactive. So over a period of time, strains are selected that have a little less temperature sensitivity. When the temperature drops in the autumn, which you can see here, then, because the virus is less temperature sensitive at this time of year, it immediately becomes more active, and you get this rapid peak, which is often seen in the autumn, where the number of people with colds and flu increases rapidly. By winter, a lot of people are ill with colds and flu, and that means that they're immobilized, so they're not spreading the virus very much. So now the virus needs to become more temperature sensitive because it needs to moderate its activity. And then, when the temperature starts rising in spring, the number of colds falls off. Now, if you compare the temperature at Christmas that year with the temperature at the end of March, the temperature was about the same. But at Christmas, the number of people with symptoms was increasing very rapidly, whereas by the end of March, it was falling off very quickly. Now, I suggest this is partly explained by a harvesting effect, which I'll come to later, but also partly by a change in the virus. There was a very interesting paper published last year in 2019 by a group at Columbia University. And what they did is they took samples from people in New York City over a period of a year and a half, and they looked for the presence of respiratory viruses in the samples. And here they've plotted the proportion of the people who have tested positive for several different viruses that showed symptoms. And you can see that an awful lot of people who were carrying one of the viruses were not symptomatic. So at one extreme, in the case of human metapneumovirus, actually about 70% of the people who were carrying the virus had symptoms. But with other viruses such as influenza, only 50% of the people carrying the virus showed symptoms. And then with many others, such as rhinovirus, RSV or adenovirus, only a quarter or a third of people carrying the virus had symptoms. In the bottom graph, they're showing the results at different times of year. And you can see that they did fewer tests in summer that year, but the proportion of those tests that were positive were roughly the same in summer and winter. So as many people were carrying detectable virus in summer as in winter. So at this point we can go back to our four mechanisms that have been proposed to explain the winter seasonality of virtually all respiratory viruses. With this data we can rule out mechanism 1, that we crowd together more in winter, and mechanism 2, that the virus, or all these viruses, survive for longer outside the body as explanations of seasonality. Because we don't need to explain why transmission is more efficient in winter, because it's not. As many people are carrying respiratory viruses in summer as in winter, so mechanisms that propose that transmission is more efficient in winter are ruled out or at least made completely unnecessary by this data. So that leaves M3, that our immune systems are somehow weaker in winter, and M4, that these viruses are temperature sensitive. Now, there are two problems for M3. First, as I said before, we have to explain why our immune systems should be better in summer in temperate regions than they are in the tropics. And second, there were a lot of experiments that were carried out in the 1950s and 60s where people were inoculated with cold viruses and then they were either chilled or kept warm. The people who were chilled were no more likely to get colds. Now, I haven't got time to go into this in detail here, but it's discussed for the non-scientist in my blog and in my 2016 review in Medical Hypotheses. So this leaves M4, which I think is the main driver of seasonality. I'm not saying that the others have no effect, especially M3. I'm just saying that they are not the main reason why we get more colds in winter than summer, caused by all these very, very diverse viruses. Now, I should emphasize that there's really nothing wrong with M4. It's not particularly outlandish, and it's based on principles that everyone accepts. Its main problem is that it goes against scientific dogma, because it goes against what biologists were taught at school. But this dogma was established in the 1950s, which was just a few years after the discovery of the DNA double helix, and molecular biology tools were very primitive. In fact, the scientists who did the experiments in many cases didn't know what virus they were working with. They just called it a cold virus or the pedigree strain. There was nothing wrong with the experiments, but I think they were misinterpreted. So we're in a very strange situation. So how does this affect the progress of colds and flu epidemics? Well, there seems to be a harvesting mechanism. In other words, the virus is present before symptoms develop, as the Columbia group showed, but when the temperature drops, people are more likely to get the symptoms of colds and flu. And I'm going to show you here an example by Professor Van Lochem, who was a professor of hygiene at the University of Amsterdam in 1925-1926, and he carried out a huge survey of people from all over the Netherlands, where participants sent him letters every week reporting how many people in their households had got colds and flu. And at the bottom of the graph, the black and white dotted and continuous lines show Van Lochem's data, showing the number of people with colds and flu in 7 different regions of the Netherlands. I've added in the coloured lines, and they show the temperatures reported by 5 weather stations in the Netherlands at that time. Again, I've plotted high temperature at the bottom of the graph and low temperature at the top. And what you can see is this extraordinary synchronization between the number of people who have colds and flu and temperature. So every time the temperature drops, the number of people with colds and flu rapidly increases. Up until mid-January, the synchronization is very good. After that, the synchronization is less clear, although it's still there. And you can imagine that probably the harvesting effect is less pronounced when the temperature is rising in spring. But what's really quite extraordinary is how people developed colds and flu all over the Netherlands at the same time, as soon as the temperature changed. So there doesn't seem to be increased transmission of the illness with low temperature. Instead, there seems to be a harvesting mechanism where what were at first commensal microbes, as Van Lochem put it, are converted into pathogens causing parasitic infections. Now, this weighs against M1 and M2, which are both mechanisms that are based on increased transmission of the virus at low temperatures. But you can see that within 1 or 2 weeks, people all over Holland are responding in a very synchronized way. Now, this is another very interesting paper. This was published in 1997 by a group of scientists called the Euro-Winter Group. And they used market research techniques in 8 regions of Europe, from northern Finland to Athens, where they looked at what they called personal cold exposure factors. And they looked at correlations between what people wore and how they behaved, and the chance that they would die of respiratory illness. Now, I've put their data on the left-hand side of the slide, and I've plotted the same data on the right-hand side. So, looking at the graph, on the right-hand side of the graph are factors that appear to have no significance, while on the left-hand side there are factors that are highly significant, and the most meaningful data is on the left. At the top of the graph are factors that are correlated with an increased chance of dying of respiratory illness, and at the bottom are things that seem to be protective. So you can see here that certain activities appear to be very dangerous. Shivering outside or being stationary for more than 2 minutes outside both seem to be very dangerous, whereas taking exercise outdoors sufficient to cause sweating is, or appear to be, because we're dealing here with correlations, highly protective. This is another very interesting paper. This was published in 2015, and it shows where different strains of influenza came from. So the authors looked at 4 different strains of influenza, 2 influenza B strains and 2 influenza A strains, and they looked at where those viruses had come from in the world. So they divided the world into 9 different regions, and they're showing where the viruses were 1, 2 and 3 years before the present. And you can see, if you look carefully, that the influenza is more likely to move from hot places to cold places than in the other direction. So if you look at Europe, for example, a large proportion of the 4 different types were in hot places a year before. In the case of H3N2, roughly 50% of the strains came from hot places just 1 year earlier. And it's also interesting to look at North and South China. 3 types of influenza were more likely to go from South China to North China than in the other direction, and in the last one, influenza B Victoria strain, there was still a significant number of strains going from south to north, although slightly more going from north to south. Generally, it seems to be the case that respiratory viruses move from hotter places to colder places, and this agrees with the TDVT hypothesis, because the strains in the tropics are in fact likely to be intrinsically more virulent because they have less temperature sensitivity, so they're more likely to go to people's lungs if they happen to arrive in temperate places. Now, there's a lot of biochemistry backing this up, which I haven't got time to go into, but I'm going to mention one aspect. Influenza is the most studied respiratory virus, and it has this temperature-dependent switch, where it switches from making protein to making viral RNA when the temperature drops. So at 42 degrees, the virus is mainly making protein, but when the temperature drops down to 37 or 34 degrees, then it starts making viral RNA, which is the genomic RNA of the virus. Now, bear in mind that these are strains of influenza that have often been in laboratories for decades, so their temperature sensitivity is very likely to have drifted as natural selection adapts them to their new environment, even though, of course, it's an artificial environment. If you're skeptical about the idea that viruses often moderate their activity, then one thing to think about is what happens when viruses jump species barriers. So I've listed here several viruses that have quite recently jumped into humans, and all of them are pretty virulent. They all tend to kill people, whereas the well-established human viruses that have been in humans for much longer tend to be much less aggressive. So the implication is that a well-adapted virus is less aggressive. Now, people will say that this is because the human race has evolved immunity to, for example, rhinovirus or adenovirus. And there may well be some truth in that, but well-established human viruses very rarely become like, say, Ebola, even in young children. And remember that Ebola is not well adapted to humans. The hemorrhagic fevers are particularly interesting. These are viruses that people occasionally acquire from rodents and other mammals, sometimes from ticks, and they cause you to bleed. So Ebola is a hemorrhagic fever. These viruses can be a very serious problem, for example, in the tropics or in Latin America. Normally they don't spread from person to person, although they do occasionally. But there are many, many different viruses that cause these illnesses, and there are at least 5 major categories of unrelated or distantly related viruses that cause hemorrhagic fever. So it seems that if a poorly adaptive virus takes hold and begins to replicate in your cells, it quite often rapidly mutates into a form that grows quickly, and then it's very likely to kill you. So, how much temperature sensitivity has CoV-2 retained? Well, it quite often goes to the lungs, which implies that it's not as temperature sensitive as well-established human coronaviruses and other human respiratory viruses. But on the other hand, sometimes it doesn't. So it sometimes causes asymptomatic infections or infections in the nose and throat, which implies that it has retained some temperature sensitivity. There's also quite an interesting phenomenon called COVID toes. Now these are basically chilblains, and what seems to be happening is that when the virus gets into the bloodstream, it tends to settle in the coldest parts of the body, and obviously the toes, as well as the nose and throat, tend to be quite cold. So that's consistent with the idea that it's retained some temperature sensitivity. So what practical suggestions can be made on the basis of this way of looking at respiratory illness? Well, one implication is that these viruses have lots of different strains with just very slightly different degrees of temperature sensitivity. And one reason that this isn't noticed is that scientists tend to focus on the proteins on the surface of the virus, and these have to change in quite big jumps, so you can only change one amino acid at a time. So new strains in this sense are quite rare. Scientists don't generally focus on things like the secondary structure of the RNA of the virus, which might control temperature sensitivity, and that can change gradually in very small steps. So, if we believe that the virus can change in small steps, one suggestion is, when we're running test, trace and isolate programs, we should focus mainly on the chains of infection that cause severe illness. So, for example, with this person here, if he got sick with COVID, you might very aggressively trace and isolate the people who have been in contact with him or were in contact with others who were in contact with him. Whereas this other person up here has a mild form of the disease, and we wouldn't focus on this chain as much. Another very simple experiment that could be done straight away would be to investigate the best temperatures for isolating CoV-2 in tissue cultures. So the scientific literature says that some respiratory viruses are easier to isolate at 33 degrees centigrade, whereas others are easier to isolate at 37. Now this is interesting because why should any of them in the first place prefer to grow below normal body temperature? A recent example was a paper in the journal Nature published this year by Wolfel and his colleagues, and they used 37 degrees to isolate CoV-2. But it would be interesting to find out more systematically what would be the optimum temperature. And are there differences between viruses isolated in, for example, the nose and the lungs? Wolfel and colleagues found that there were actually independent lineages of CoV-2 replicating in the throat and lungs of one particular patient. So it'd be interesting to see what the temperature sensitivities of these two strains were. It would also be very interesting to see if the temperature sensitivity of CoV-2 is changing over time. Now, I can make lots of other suggestions for research, which you'll find, for example, on my blog, Old Wives and Virologists, and there's a note there about suggestions for research into respiratory viruses in general and CoV-2 in particular, and, for example, focusing on the secondary structure of the virus's RNA, which I showed you before. Because, in fact, CoV-2 and influenza and many other respiratory viruses have this secondary structure in their RNA, which is conserved, and that means it must be doing something. And in fact, in the coronaviruses, including CoV-2 and SARS, there is this structure called the S2M structure, and this is actually tertiary structure, which means that it has a very well-defined 3D structure. And it'd be very interesting to find out if it was actually involved in any sort of temperature response in coronaviruses. Now, I find that scientists very often don't understand the TDVT hypothesis, and I'm not sure why. On the other hand, non-scientists usually do, so I'm going to quote my friend Brian when he summarised the main hypothesis. So this is what he said. In order to replicate, a virus moves from host to host. This requires a transmission method, sneezing, which will fail if the host dies or is too ill to socialise. Moderate symptoms and a long incubation period are thus ideal. The virus accomplishes this by being sensitive to temperature. The colder temperatures are in the mouth and nose, which is where transmission through sneezing will happen. To reproduce at higher temperatures, such as in the lungs, would incapacitate the host and so prevent transmission. However, the problem with CoV-2 is that it seems not to be sensitive, or as sensitive to temperature, so getting into more lungs. The seasonal pattern of respiratory viruses is a consequence of this. As temperatures rise in the air, and thus in the mouth and nose, The virus tends not to replicate in any part of the body. Is that it? To which I replied, Brian, you have absolutely got it. These concepts suggest new approaches to research into respiratory viruses, focusing on temperature and temperature changes. I propose testing the ideas at many different levels, including looking at the responses of individuals to chilling, making greater use of temperature in the biochemistry wet lab, and modelling the effect of temperature on the conformation of viral RNA. And that's the end of my talk, so thank you very much for listening. And if you want to find out more about the TDVT hypothesis, then you can read my paper, which was published in Medical Hypotheses in 2016, or look at my blog, which is at oldwivesandvirologists.blog. Thank you very much.